Thank you so much to all the organizers of Bionano Symposium 2026 for giving me the opportunity to present my work. My name is Debopriya Chakraborty, and I'm a molecular diagnostics fellow in the Clinical Genomics and Advanced Technology Laboratory at Dartmouth-Hitchcock Medical Center. And today I will be talking about an evaluation study on recurrent pregnancy loss, which I will henceforth refer to as RPL, using optical genome mapping or OGM integrated with gene-specific structural variant profiling. I do not have any conflicts of interest to disclose. To begin with, as per ACOG, the definition of recurrent pregnancy loss or RPL is the loss of two or more pregnancies as confirmed by ultrasonography or histopathology in pregnant women prior to 10 to 24 weeks of gestation. The testing for RPL involves ultrasonography, maternal screening and autoimmune diseases and hormone abnormalities, or parental karyotyping for parents at risk. Some of the factors which predispose an individual to RPL are parental chromosomal abnormalities, structural uterine abnormalities, and endocrine disorders. Looking at the epidemiology, approximately 1 to 4% of pregnant women in the US and Europe have RPL, and there might be some discrepancies in these statistics based on the number of women who are actually reporting the pregnancy losses. The management for RPL involves identification of the factors, intervention with supporting evidence, and lifestyle alterations before pregnancy. Some of the factors that predispose an individual to RPL involves previous pregnancy losses, family history of pregnancy loss, weight or BMI, occupational and environmental exposures, stress, maternal and paternal age, as well as habits such as smoking, alcohol, or caffeine intake. Looking at the demographics, approximately 10 to 15% of clinically recognized pregnancies and almost 30% of all pregnancies can result in spontaneous abortions. 5% of females experience 2 or more consecutive miscarriages, which can result in RPL. The risk of developing RPL usually increases with each miscarriage. And it has been shown that approximately 50 to 75% of RPL cases have idiopathic causes. Now, looking at some of the maternal and paternal specific factors that can lead to RPL, age and chromosomal abnormalities are contributed by both the parents as a reason for pregnancy loss. There are certain factors such as Y chromosome deletions and DNA fragmentation which are male specific, while others such as uterine abnormalities, cervical incompetence, or hormonal anomalies that are contributed by the mother and can lead to pregnancy loss. Looking at some of the signaling pathways that might be involved in RPL pathogenesis, it has been shown that IL-6, JAK-STAT pathway, PI3K AKT signaling, MAP kinase, and NF-gamma B that results in immune response, invasion, migration, apoptosis, proliferation, and angiogenesis downstream might be affected in RPL. Aberrant expression or repression of these pathways are usually result in recurrent pregnancy loss. Some of the workflows for detection of chromosomal abnormalities are the most common one is karyotyping, which can detect structural changes and aneuploidies, FISH or fluorescent in situ hybridization that can detect microdeletions and microduplications, chromosomal microarray or CMA that can detect copy number variants, and then sequencing technologies can detect single nucleotide variants, small indels, and some copy number variants. And the most recent technology, OGM, which is able to detect structural variants across the genome and also detect certain cryptic rearrangements, which are undetected by the other technologies. Coming to optical genome mapping, as I mentioned, it detects structural changes that are limited by karyotyping and CMA analysis. It has the ability to detect balanced reciprocal translocations, which occur in approximately 2 to 8% of all RPL cases. It is recognized that in referrals of recurrent miscarriages, approximately 25% of chromosomal abnormalities go undetected, and this could be detected by optical genome mapping. It is a cost-effective and complementary approach to traditional cytogenetics methods. The only caveat or limitation of this technology is that it is less sensitive in detecting balanced Robertsonian translocations. The objective of my study is it's an exploration study, so I want to explore a workflow that uses a list of curated genes for RPL and want to evaluate the performance of this gene list against certain cases that we analyzed by OGM. And these were also complemented with other assays such as karyotyping and chromosomal microarray. To start with, we did a literature review of the genes that are potentially associated with recurrent pregnancy loss and other infertility conditions by PubMed search. The gene list was pre-curated by filtering out all those genes which had poor functional significance or had unknown RPL associations, especially genes that encoded microRNAs or HLA antigens. Genes with weak or indirect associations, such as differentially expressed genes, were also filtered out from our analysis. We made the final list by including genes that had an OMIM phenotype, and we also highlighted the ones with infertility associations in OMIM. The final list of genes prepared post-filtering was then converted into a region bed file based on the GRCH38 coordinates. So this is a snapshot of the gene list that we curated, and this needs to be further refined. However, this is a preliminary set of 238 genes, and the ones which are marked in red have OMIM infertility associations. Now, next, since I talked about certain signaling pathways that can be altered in recurrent pregnancy loss, we wanted to see whether these genes are involved in some signaling pathways. And for that, we used this online pathway analysis tool called Reactome, and we put our 238 genes into that platform, and we were able to identify 10 significantly associated signaling pathways in relation to these 238 genes, among which the pathways involving extracellular matrix organization, signaling by interleukins, and insulin receptor signaling has been implicated in early fetal development and reproductive biology, and might have implications in recurrent pregnancy loss, according to literature studies. Now, looking at our case demographics, we had 17 cases in total, and this was a research cohort where we used residual samples from our already tested cases, and 65% of our individuals were female. When we did an age distribution analysis, we found that most of these individuals were within 30 to 39 year of age range. Now, looking more into these cases, we tabulated the cases based on the karyotype that were done for some of these cases, and also the indication for testing. The indication included a broad range from individuals having a family history of genetic disease, infertility conditions other than recurrent pregnancy loss, and certain chromosomal aberrations such as balanced and unbalanced translocations, as well as those individuals who had recurrent miscarriages or recurrent pregnancy losses. So looking into the first case that we want to dig deep into, case 6, here a karyotype analysis was performed, and this was a female with a balanced reciprocal translocation involving chromosome 611. When microarray was done on this individual, the results were normal, and as microarray has the limitation of not being able to detect balanced translocations. However, as a proof of principle, when we performed OGM, we were able to recapitulate the karyotype results and were able to detect the 611 translocation, as you can see on the circus plot here. Further, alignment of the molecules on the genome map confirmed this translocation, and it was at the exact location as determined by karyotyping. And the two genes that were involved in the region of translocation were SBF2 and USP49, which resulted in a putative fusion. However, these genes were not a part of the 238 gene list that we prepared. So we wanted to further see what the function of these genes are. So when we looked at SBF2, we saw that it causes autosomal recessive condition that is characterized by demyelinating neuropathy and early onset muscle weakness. USP49 is not associated with any Mendelian disease phenotype. However, it encodes an enzyme that causes histone modification and is involved in regulation of gene expression, suggesting that the offspring might present with certain congenital anomalies. The second case that we looked into was a female with a history of chromosomal abnormality leading to a miscarriage. When we looked at the product of conception, we saw that there were multiple fetal anomalies as seen on ultrasonogram. When we looked at the microarray results on the product of conception, there were certain copy number variants that were detected. However, no translocations were detected by microarray. Looking at the OGM results, we were able to rule out translocation by looking at the circus plot. However, there was one of our RPL candidate gene FLT4, which was detected in a region of gain on chromosome 5. This FLT4 gene is associated with autosomal dominant conditions such as congenital heart defects and lymphatic malformation. It has been shown to play a role in angiogenesis and has the potential in placental development. So disruption in this gene can result in recurrent miscarriages. And there has been studies involving sequencing that had shown pathogenic variants in this gene being associated with recurrent pregnancy loss. The third case we looked into was again a female with a history of recurrent pregnancy loss. She had 3 miscarriages. Again, OGM was able to rule out any translocation as you can see in the circus plot. However, again, on chromosome 4, we were able to detect a deletion encompassing one of our RPL candidate genes, FRAS1. FRAS1 is associated with a rare genetic disorder, an autosomal recessive condition that can cause certain birth defects like kidney agenesis and can manifest into recurrent pregnancy loss. And it has been shown through evidence that in cases of recurrent pregnancy loss for consanguineous couples who have variants or pathogenic variants in the FRAS1 gene. We were also able to detect another deletion on chromosome 16, which encompassed another gene in our RPL gene list, ABCC6. This gene is associated with a connective tissue disorder, autosomal dominant condition. And even though variants of this gene typically do not cause high rates of pregnancy loss, placental calcification can occur as a result, and there can rarely be certain growth issues. There is evidence which suggests that there are certain cases with idiopathic recurrent pregnancy losses where discovery of pathogenic variants such as ABCC6 has been detected. To summarize, we analyzed in total 17 cases using optical genome mapping. A subset of these cases were complemented with karyotyping and chromosomal microarray in individuals who either had recurrent pregnancy loss, infertility, or a family history of chromosomal abnormalities such as genetic disorders. We were able to demonstrate in these cases that OGM was able to rule in or rule out chromosomal translocations implicated in RPL. And we were also able to present some example findings in which structural variants and copy number alterations were identified in RPL candidate genes, such as FLT4, FRAS1, ABCC6, which are involved in some way with placental development and fetal development. The only caveat we had with this study is that we had limited number of cases. There were only 17 cases, but we are trying to expand our cohort in the future. With that, I would like to acknowledge all the members of the CGAT lab, cytogenetics and molecular section staff, Dr. Wahab Khan for his support and guidance in this project, our genetic counselors, Devin and Amy, and the organizers of Bionano Symposium for giving me the opportunity to present today. Thank you, and I can take any questions.